Ladies and gentlemen, welcome back to the PrimateCast. I'm your host, Andrew McIntosh, and unfortunately, my co-host, Chris Martin, won't be able to join us today because he's off preparing for a week-long trip to Borneo, where he'll be going next week to see wild orangutans. But he will be back for the next podcast, in which we speak with GIS specialist Dr. Janet Nakoni. But first, in today's interview, we're going to be going back to November of last year, when we sat down with Dr. Lisa Jones-Engel. She's Senior Research Scientist at the Washington National Primate Research Center and also a lecturer at the University of Washington. Now, Lisa was in Japan for a workshop put on by faculty here at the Primate Research Institute, and Lisa herself was invited because of her work on many issues surrounding commensalism between humans and primates, specifically in the bi-directional disease transmission between both humans and non-human primates. So in this interview, we'll be talking about some of her work, notably concerning some charismatic diseases such as tuberculosis, as well as the role of Asia in the world of emerging infectious diseases. Now we're going to begin talking to Lisa about her work at the biomedical centers in the US, the evolution of her research, and towards the end, interestingly, about her take on the role of primatologists in emerging infectious disease research. But first, here's Lisa talking about how she came to be working at the WNPRC.
You know, I've had this weird series of trainings that have actually had me in primate centers a lot, not doing any biomedical research, but I started out at LEMSIP, which is unfortunately named the Laboratory for Experimental Medicine and Surgery in Primates, which was attached to NYU in upstate New York. And I was the nursery technician, so I got to care for a bunch of baby chimps, and that was back at the time when you could actually take the chimps home. So I was in LEMSIP at that point. I did some work down in Yerkes, again, and this was more psychological, Bailey's testing in the infant lab there as well. And then I was recruited to come up to the Primate Center, and as an anthropologist, when someone offers you a job, you say yes, basically. And so I went up there and did a postdoc that was part kind of basic laboratory training and some basic virology. And I did that for a little while, then actually stepped out of my postdoc because there was this interesting thing that you're only allowed to, you can only PI your grants if you have a certain standing, and postdocs don't have that standing.
Right.
So that was news to me, so I didn't. And one thing led to another, and somehow 10 years have gone by, and I'm still at the Primate Center, which is very odd for someone like myself as a field-based researcher. People say, what are you doing at a biomedical facility? And it's a good question. It's a very good question, except that it has really, because I do so much infectious disease work now, being in a facility like this really gives me access to, if I have a question about SRV, I can call my diagnostic lab. If I need some help reminding or refreshing on placing a TST in a monkey's eyelid, I can talk to veterinary staff and go down, and when they're doing routine testing, I can kind of step in there with that. So it's, in many ways, it's been ideal. But it can be very hard to rationalize that, occupying that space in what I do.
I want to touch a little bit more on that because you are a primatologist, came through the anthropology department, and now, of course, most well known for work in infectious diseases involving primates, particularly zoonotic diseases or anthropozoonotic diseases. So how did you make that transition? At what point, when you were coming through your degree programs, for example, did you decide that, okay, I need to be interested in disease here, or I am interested in disease?
You know, the answer to this is somewhat embarrassing, but it's in part the person that I'm married to. So my husband is an MD epidemiologist, Gregory Engel, and we've been together for 27 years. And so for a long time, I was trying to figure out how to meld my interest in primates, primate behavior, primate ecology, primate conservation, and then this guy who was doing medicine, and one day it finally all came together to me, because I'd always been interested in herpes B, actually. It had never made sense to me, having spent my early years in Asia, where all these monkeys are around and people are around all these monkeys, and no one was dropping dead from this virus. But I had known from Lemsip and Yerkes that the biomedical community, the laboratory community, is terrified of this virus. And I couldn't, it doesn't hold up. That fear doesn't hold up out in the wild because it's not the same level of pathogenicity isn't evidenced there. And so I'd always thought about that, and one day it was like an epiphany. I thought, well, so I have this whole monkey perspective and this anthropological human primate interface thing. My husband has the medical side of it. I had spent a lot of time around a lot of veterinarians and kind of been brought up by primate veterinarians. It suddenly seemed to me the logical thing to do was to meld all those different skill sets into this one project. And I realize how lucky I am that, if I have a question about, Gregory, what's the difference between H3N2 and H5N1? I don't actually have to crack a book. I can simply, when he walks in the house, and sweetheart, explain this one to me. Or I still don't quite understand that sensitivity specificity thing. Can you break that down for me? And that's been critical in actually bringing this project to the level it is now.
And so yesterday when you gave a talk, I think you were talking about three main say diseases or pathogens or infectious agents. So can you just give us a rundown of that research?
I will, and thank you, because this is my soapbox right now. Tuberculosis in non-human primates, both in wild populations and in captive laboratory populations. Enteroviruses, which are in the family of the coronaviruses. Polio is the most widely recognized. And the third one is the simian foamy virus. It's got this great name, foamy, and people always ask me, does that mean you start foaming at the mouth when you get this virus? No, it doesn't mean foam at the mouth, but it does. It actually kind of describes what the cells look like in culture. The TB one is the one that we've really been focusing on a lot. We've published 3 papers in the last 4 months on tuberculosis. It's one of the things that we were interested in very early on was markers for transmission from humans to primates. And the original dogma was that non-human primates, macaques, are exquisitely sensitive to tuberculosis. That, you just wave a few bacilli in front of them and they just drop over dead. In laboratories, outbreaks of TB are the things of nightmare. It used to be that if an animal became TST positive, that's that little tuberculin skin test in their eyelid, the whole room would be quarantined, and often the entire room sacrificed in an effort to stop this bug in its tracks. And again, this kind of goes back to the herpes B thing for me. The dogma in laboratories and reality on the field, or in the field, they're really at odds. So we know there's tremendously high prevalence and certainly incidence of tuberculosis in Asia. And there are lots of macaques running around in Asia. These macaques are not dropping dead from TB. Why is that? The problem is trying to place a TST, a tuberculin skin test, in a macaque's eyelid and then follow them at 24, 48, and 72 hours is challenging. We've done it a few times, but it's a really challenging thing. So we wanted to come up with another way that would allow us to see if we could detect the presence of tuberculosis. And the nice thing about being a primatologist, I've found when I've doing this over the years, is that because I generally I'm not so steeped in microbiology or in virology or any of the other disciplines, I actually don't get kind of worked up over why something won't work. So I thought, I was a graduate student with Alicia Wilbur at the time, and she was using a swab of the mouths of this indigenous group in Paraguay, the Ache. She was swabbing their mouths and then doing a DNA test, trying to detect the presence of the Mycobacterium tuberculosis DNA in their mouths. She was also a biological anthropologist, and her rationale was that TB basically enters and exits the body through the oral cavity. It's a respiratory pathogen, you can inhale it. We think that human-to-human transmission comes from close contact and being exposed by these respiratory droplets. So it always seemed logical to me that, well, then why wouldn't you be able to find some of the organism in the mouth? And so with her help, we started swabbing monkey mouths. When we would sample these animals in these countries, we'd swab their mouths, take the swabs back in this very simple lysis buffer that you can keep at room temperature, which is important, listeners, because you can do this in your own countries, in your own field sites. Because you can keep it at room temperature for a long time. Taking it back, and then the laboratory, that's a kind of very straightforward DNA extraction technique. And then there's this little fragment called IS6110, which is diagnostic for this Mycobacterium tuberculosis complex. You only find this fragment in the MTBC complex, or the MTB complex. So we found that in populations where the prevalence in the human population for TB was very high, we saw this very nice epidemiologically consistent pattern in the primates. So for example, in Sulawesi, which has the highest prevalence of tuberculosis throughout all of Indonesia, and Indonesia is already high to begin with, but northern Sulawesi is the highest. In our pet monkeys there, we found, I think it's 70% of those animals, we could detect the MTB DNA in their mouths. Similarly, in a temple site in Bali where there was an outbreak of tuberculosis, so we actually had both TST tests and our own OSP, this oral swab PCR. We could correlate the two. We found a very high prevalence. In a place like Gibraltar, where you have the Sylvanus, where the prevalence of TB in the human population is virtually non-existent, we couldn't detect any MTB DNA in the mouths of those monkeys. So for us, it was like, hey, this is a great test to use. So we went to some of the alpha males in the human TB world at that time and said, look what we just found. What do you think this is? And they just got all bushy and puffed up and said, no, this can't be. Because if a monkey has TB, it will be dead. All of the data says monkey has TB, it's dead. Were your monkeys sick? Were your monkeys dying? I'm like, no. Well, then you're obviously picking up some other mycobacteria. Like, but the American Thoracic Society says IS6110 is diagnostic for MTBC. It's what you all use in humans. No, this can't be. This simply can't be. And so we actually sat on these data for years. We would keep trying to farm it out there a little bit. It gets knocked back down by the human TB world, and we kind of back up a little bit. But we finally got to the point where, I'm pretty sure we're right on this one. And then we've had recently a nice way of looking at this, in that there was a case of tuberculosis, quote unquote, atypical tuberculosis in our colony. We just published this one in Emerging Microbes and Infections a couple of weeks ago. So this was an animal that was infected with SHIV, which is that human chimeric HIV virus, or simian chimeric HIV virus. Had weight loss, diarrhea, and so when the pathologist opened the animal up on necropsy, they looked at it and they saw these granulomas. They said, well, this animal's part of an SPF colony, specific pathogen-free colony, which means it doesn't have tuberculosis. So this must be this atypical mycobacteria tuberculosis that you see as this normal progression to SAIDS. Goes the animal back up. And we knew that there had recently been an outbreak of tuberculosis in the colony, a case of tuberculosis in the colony. So we actually asked the staff, the veterinary staff, for swabs, necropsy swabs from that index animal, plus contacts of that animal in that room while they were still alive. And then we got swabs from, as those animals came to the end of the study and were euthanized, we got swabs from them at necropsy. And lo and behold, in both the animals that were alive and that shared that room with the index case while the index case was alive, we could detect the MTBC DNA in their mouths. And on necropsy, in the tissues of those animals, we could detect this MTBC DNA in their mouths. And yet, pathology would say, no, there's no presence of mycobacteria in these animals. And so for us, we thought, wow, this is really important, because if you have an animal, and let me back off. The reason the primate centers exist, I think, is for the primate model. These biomedical facilities use the non-human primate to model infectious disease in a lot of cases. We use it for neurology studies, we use them for colorblind studies, but they've primarily been models for biomedical disease. And your model is really only as good as it is characterized. So if you're infecting an animal with SHIV, and you're looking at immunological parameters that you can follow in that animal because you think it's infected with SHIV, but if you don't know that animal's actually infected with tuberculosis, how do you know what you're seeing in that animal is a result of the infection with SHIV or the vaccination, and not this kind of synergistic interaction with another pathogen? These papers have not won us any friends at the Primate Center. Now, we understand why, and we will continue to push this forward, but it's, the thing about TB is it's really old. The genome was 2.5, 3 million years old. It's been around a long time. We used to think it was a crowd disease. Hell, we used to think that human TB came from bovine TB. So we're learning a lot as it goes on, but this pathogen's been around a lot longer than there were ever crowds of Homo. It's an obligate pathogen. It has to have a host in order to exist. It can't just hang out in the soil. So there wasn't a lot of human-like Homo ancestors 2.5 million years ago. There were a lot of primates 2.5 million years ago. And so for us, it's given our training, it's easy to think evolutionarily how this pathogen could have adapted with these different primate populations. And that coevolution means that you're very likely to see different patterns or different manifestations of disease. It's been a challenge, though, trying to get people to buy into this one, but so it's steady. The data will just keep coming out, and dogma eventually falls. In the face and weight of data, dogma falls, but you just have to keep piling on the data.
So I imagine the reaction to this, in another sense, was, or part of the interest would have been in, okay, so now you have a reservoir for human tuberculosis as well in the macaque populations, and tuberculosis in other animal populations are currently in the news as well, in England, for example, where they have they have their culling scheme with the badgers to prevent bovine tuberculosis. But so what is the situation like currently then with the macaque TB?
You're talking about the wild populations?
The wild populations that you were sampling from and found this using the oral swabs. Yeah.
I think we're so early on with this that I think one of the call to primatologists has to be, here is a way for us to start looking at this pathogen, and this one is a pathogen, in primate populations. This is a fairly easy test to administer. You don't actually have to anesthetize the animal. You can use these chew swabs, these flavored swabs, toss them out, collect it. We're actually working right now on developing assays that allow us to strain type the tuberculosis that we're finding, because if what we're looking at is patterns of transmission, you need something finer than to just say this is part of the MTBC. We want to know, is this the Beijing strain? Is this an animal strain? Is this strains we're more likely to see in East Asia versus strains that we're seeing in Africa? So we need a lot more data. We actually need the primate community to kind of step in and come forward with that. But there are lots of strains out there, and there's evidence to suggest that some of the more ancient strains are less likely to cause disease. We don't know yet really what level of pathology is happening in these populations. Certainly that outbreak that was in Indonesia years ago, those animals got pretty sick. Could that be that there was a particular strain of tuberculosis that was highly pathogenic for macaques? Yes. Could it be that those macaques were infected with something else that immunosuppressed them? Possibly. There's not enough research out there being done on this yet to know.
Okay, well, we'll definitely look forward to more from you then in that regard. So enteroviruses.
Enteroviruses are fun because they're fecal-oral. Kind of the upshot of this one was we were looking in Bangladesh. The CDC had been in mid-2000s screening human populations in Bangladesh, looking for, as part of the polio screening projects, any viruses that were associated with significant pathogenicity in humans. And so they found in a handful of sick human kids these viruses that when they strain typed them actually were more closely related to a simian enterovirus that had been isolated back in the 1960s. So remember in the 40s, 50s, and 60s, 70s, we were bringing lots of macaques from the subcontinent over into the US for biomedical research. Mid-70s, that stopped. IPPL managed to kind of shut that down. We haven't really seen any import from Indian origin macaques since then. But there were lots of these macaques in facilities in the 60s and 70s. And at some point, someone found this SV, EGADS, why can I not come up with SV46, SV41, the simian virus, some number. Anyway, so the CDC found that this EV76 and 89 and 90 and 91 that they found in these kids was most closely related on a phylogenetic tree to the simian virus. So they came to me and said, Lisa, do you have any idea what's happening with these viruses in monkeys in Bangladesh? And I'm like, I don't know, but let's go looking. So we did this study where we looked at like 750 fecal samples from macaques all over Bangladesh. And the upshot of it was, and it still shocks me when I think about this, any of the macaques that were synanthropic, and a synanthropic macaque is basically, a synanthropic animal is an animal who thrives in a human-altered environment. Macaques are really good at doing that. When humans change the environment, macaques, they jump right in there and their numbers actually increase. They're great at that. Any macaque that we sampled that was in a close relationship with a human, when we looked at their enteroviruses looked most like human enteroviruses. We couldn't find a monkey virus in a monkey that was if it was in association with humans. In order to find monkey enteroviruses in Bangladesh, we had to go to the Dhaka Zoo. And that was the only place that we could find monkey enteroviruses were in populations of macaques that were in these captive settings. We found a handful of human enteroviruses in those animals as well. So if you're a monkey and you're living with humans, your viruses look most like that human population. Your enteroviruses look like that human population that you're around. So the problem with the naming of viruses is often we tend to call a virus, depending on what we find it in, we call it that. So if we find a virus in pig, we call it a pig virus. We find a virus in human, it's human virus. If we find a virus in a monkey, it's a monkey virus. That really doesn't speak to, though, what is the natural reservoir for that virus. So we don't know if these highly pathogenic viruses we were seeing in the humans, because we also found those in the monkeys, were monkey viruses that had jumped the species barrier at some time, or whether these were human viruses that the monkeys had picked up. We're back. This is the next stage of this research, is to really focus on that.
So in most of the research that you've described, particularly when you're talking about trapping like 1,000 monkeys or something like that, I imagine there's so much breadth. So geographically and in terms of the number of animals that you're looking at. So how do you make that transition then from mass sampling to really starting to understand the biological relationships between those organisms?
That is such a good question. And it's, I think for many years in my career, I was sampling in many, many different places because I wanted to picture a sense of what the landscape looked like. But I think I've come to the realization in the past couple of years that it's important to tunnel down much more directly. So that's why. So we're actually in our sixth year in Bangladesh on this evolution and emergence of these simian retroviruses, or these enteroviruses. And I think to get at some of these questions of how viruses emerge and recombine, and the effect of this anthropogenic effect, you have to be willing to kind of set up in a country for a while and really sit down. Longitudinal sampling is absolutely critical for that. I'll give you an example from Singapore. So years ago I published a paper that said we found all these antibodies to measles in the Nepal monkey population. In the Singapore population, we didn't find any antibodies to measles. And so I went off and wrote this thing about a risk analysis and risk assessment, and why we weren't, why that could be the case in Singapore, that we weren't seeing it because the humans are vaccinated and better sanitation, blah, 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 and why in Nepal all these monkeys had antibodies to it. And it seemed to me like a neat little story, except that as I've continued to sample in Singapore, I've seen that there's this one sampling period where all of a sudden, boom, I got this absolute hit of young animals that were antibody positive for measles. And if I hadn't been doing this longitudinal sampling, I could very easily have missed that little flare that happened, because in subsequent years, it's gone. I haven't seen it there. Being able to sample the same monkey twice is virtually impossible. Monkeys just, even though I chip all my animals, I think I have a grand total of 7 animals out of 1,000 plus that I've ever caught twice. So it's really important to sample broadly and sample longitudinally, or you're going to miss these viruses, these pathogens that may not be frequent, but they give you a real sense of what could be going on in these populations. So that's why for us in Bangladesh, it's been very important that we've been going back every year and we're getting longitudinal samples from these animals. So you see how viruses are being introduced, you're seeing how there's actually changes within a virus, within a population over time, but you have to be willing to kind of set up shop in there, and you have to have really strong in-country collaborators to make that happen.
And so all of this research that you've been doing over the past 10 years at least, and continuing to do, has happened in Asia. So you had a great point about, or a few great points about why this is the case. So maybe you could reiterate that here for us.
It's, Asia, it's not a coincidence that several of our latest emerging infectious diseases have come out of Asia. SARS, Nipah, several of the influenzas. Asia has this kind of magic combination of incredibly dense human populations, a lot of them increasingly immunocompromised with tuberculosis and HIV. It has the infrastructure that allows people and animals and goods to move very rapidly in and out of the area. It's got biodiversity hotspots. So the big thing about, I think, emerging infectious disease work is kind of these 3 Ds. You want the density of population, humans and primates. You want them distributed across a variety of different contexts of contact. And you want diversity of primate species, because if you're interested in recombination, and Asia really has all those things. Plus, Asia has this wonderful cultural and religious tradition associated with primates. Hanuman is a monkey god. The monkeys are viewed as, the monkeys today are in parts of the subcontinent are viewed as the children of Hanuman. As such, there's a reverence and a tolerance that we see for them, which isn't, you don't really have that same association with primates in really any other part of the world. Asia is where it's happening with that.
And so another point you brought up that I really enjoyed hearing and listening to was, again, as we mentioned earlier, that you are a primatologist. So what is the role of the primatologist in all of this?
Primatologists are the consummate generalists. We're not particularly, certainly there are some primatologists who are absolute specialists. Myself as a primatologist, I'm never the expert in the work that I do on my team. The virologists always know more than I do, and they're very happy to tell me that. The microbiologist, the same deal. The parasitologist, my husband often tells me he knows more than what I do about these things. But our training as primatologists and as biological anthropologists really allows us this amazing latitude. We're trained to observe and to look for interactions and look for connections between animals and people. And it's easy then to kind of open yourself up as a primatologist and look for connections between the virologist and what I do. The trick comes for the primatologist is to be able to have enough knowledge and have a vocabulary that allows you to basically translate between the different disciplines, because that's, I think, where multidisciplinary research often stumbles. Virology speak is very different than primatology speak. Bioinformatics speak, I don't even know what they're saying, but they speak it. But as a primatologist, somehow we seem to be able to reach out and to connect to each one of these different disciplines when individually they rarely are able to cross the aisle. So I think that's what makes us so critical on these teams. It's certainly, you always need the otherologist. They have the expertise, but the real power, I think, comes from the primatologist. Not just because what we know about the primates, it's what we know about people.
Well, Lisa, thank you very much for joining us here on the PrimateCast.
Thank you for having me.
You have been listening to the PrimateCast, a podcast series dedicated to the study and conservation of primates around the world. Brought to you by the Centre for International Collaboration and Advanced Studies in Primatology of the Primate Research Institute of Kyoto University. Visit us online at www.cicasp.pri.kyoto-u.ac.jp/news/podcasts and follow us on Facebook at www.facebook.com/theprimatecast and on Twitter at theprimatecast.